out as institutions were receiving funding in order to enroll individuals into longitudinal studies. And now it's shifted to really investing in ideas, novel ideas, things that can advance the science. And I think it's going to continue having more funding for even more investigators across all stages.
The state has been able to appreciate the progress that's been made. So there's been a great deal of success in bringing federal funding to Texas for Alzheimer's disease. And I think TARC has been the primary stimulus for bringing that federal funding here.
We have heard about a return on investment of at least $4 to $5 for every dollar that is put in. But there are additional aspects, and we're desperately short in most of those areas across the U.S.
The existing funds for Alzheimer's and dementia care research is phenomenal, but we can always utilize more. We could use more, we would benefit from more. So many of our efforts are limited just by the available resources given to us. And so with more resources, we could really expand our efforts, expand our reach, and be able to have that greater impact in the community.
Continued investment means progress, and until we find a cure, then we need this investment.
Please welcome Dr. Claudia Satsabal. Dr. Satsabal is an epidemiologist and associate professor at UT Health San Antonio and the Briggs Institute, where she leads the Population Neuroscience Core. Her research focuses on the lifestyle and genetic factors influencing brain aging and dementia. Currently, Dr. Satsabal is exploring the factors contributing to dementia disparities among Mexican Americans in the San Antonio Heart and Mind Study, the identification of a gut microbiome signature of Alzheimer's dementia, the validation of dementia biomarkers, and understanding the genetics of Alzheimer's disease among Hispanic and Latin populations.
All right. Thank you all. I appreciate the invitation to be here today and tell you more about our collaborative grant that we have together with Dr. Joe Petrosino at Baylor College of Medicine and myself at UT Health San Antonio, exploring gut microbiome signatures related to brain aging and Alzheimer's disease. So a little bit of a disclaimer, we are still collecting data. So what I'm going to do today is I'm going to present an overview of what we know about the microbiome, and then I'm going to shift to our progress and how this data can be valuable not only for this project, but for the whole community. All right, so what is the microbiome? And so this term has been defined as the collection of genes and genomes that our microorganisms host. And sometimes a microbiome and microbiota are used interchangeably, and I may do that in the talk. But basically, the gut has the most gut microbiota in our bodies, with almost 1 trillion microorganisms. And if you compare that to the number of cells in the human body, the relationship is almost 1-to-1. So not only that, but some people consider this as a second genome. And this is because all the microorganisms that we host have 100 to 500 times more genes than the human genome. So you can imagine that all these genetic diversity we host can influence metabolism and brain health and disease. So how do we get the microbiome in the first place? So this is pretty much influenced by the exposome. And it starts with, during conception to birth. There's some studies that suggest that maybe a healthy womb may have a little bit of microbiome. Others are not so sure about that. But really is when the baby is born that is kind of empty field so that all the microorganisms want to jump and colonize that baby. And what is very important is the way of delivery, whether it is natural or by C-section, that will influence what microbiome kind of sets in that baby, and also what type of feeding it's following. Is it breastfeeding versus formula? And later on, what are the first solid foods introduced into the diet? In this period of time, since birth and until the first 1-2 years of life, are the most plastic and are those where really a particular microbiome sets into the body. Later on, as childhood and adolescence progress, there's a little bit more stabilization of that microbiome, and it stays pretty much stable other adulthood. And once your persons reach older age, there is some research suggesting that that diversity decreases. And so in 2008, the National Institute on Health started this project to better understand the microbiome and how it influences health and disease. And it's the Human Microbiome Project. And it delivered many important findings, including kind of the mapping of all these microbiota signatures. And as you can imagine, it will depend pretty much on which part of the body that is exposed to the environment will have. So as you can see in this figure, there's, if you sample your microbiome from the skin, it has a very different distribution than, for example, that of the gut, which includes primarily bacteroidetes. And so how can we measure this? The techniques have evolved over time. At the beginning, all the studies were pretty much done in cultures, but these methods led to very low throughput, unless other techniques later developed to assess at the same time many microorganisms. But in general, the problem is that for example, bacteria you can identify will depend on whether we have the medium and whether that bacteria can stand the outside conditions like oxygen and things like that. So it was pretty limited. Later on, there were some methods that evolved to assess highly variable regions in microorganisms. So for example, in bacteria, there's this particular region, the 16S of ribosomal RNA, highly variance regions that are sequenced to better understand what are the taxonomic features of that particular microbiome. And that really improved the ability to detect the diversity of microorganisms in a person. It's relatively cheap and it's fast. It has some limitations. For example, the resolution is limited to the genus level, and there might be some limitations with low biomass. But otherwise, it's a pretty common method right now used by many labs. And then we have metagenomic sequencing, and this really has been implemented in recent years. And what the method does is that it sequences all the genetic material in that particular microbial sample. And it allows for a much more refined taxonomic resolution. However, is a little bit more costly, as you can imagine, and it depends pretty much on the libraries that are available. However, those have been improved over time, and there are new techniques to do QC and to really clean that data up. But yeah, it's a lot of data, very interesting, and that is one of the things we are doing in this project. There are other techniques that can be used to assess the virome. I'm not going to go over that today. We're focusing pretty much on bacteria and other metatranscriptomics that allow for understanding of the function of that microbiome in the host. And so, for example, once we've done the analysis, what do we get? How do we quantify this information? And so there's two things you can do with this data. One is to assess alpha diversity, and this refers to the diversity of the species, how rich, how different they are in a person, and how even they're distributed. Is it that a particular set of bacteria is just it has very low abundance versus another strain that might be more abundant, or are they evenly split? The other aspect is beta diversity, and this basically quantifies the variation between samples. And with this, you can see, okay, if we compare these two persons, we can see what is the degree of similarity or dissimilarity according to the shared species between those two. So where does the brain come in all of this? Apparently, 2,000 years ago, this code was attributed to Hippocrates, although it's hard to prove. All diseases begin in the gut. And although it's hard to really assess that, there's been over time longstanding recognition of the influence of the gut on the brain. And now we know that there is a very dynamic interaction between the gut and the brain. There are many factors influencing this dynamic communication. For example, on one end we have genetics or diet. It's quite important. As we discussed, the mode of delivery, all the environmental factors, built environment, what people are exposed to, drugs, for example, antibiotics, steroids, and things like that for allergic diseases, especially in the first years of life, seem to be very important to determine later health and behaviors like physical activity. On the other hand, this interaction can influence, for example, other behaviors like food cravings or cognition. Perhaps after a meal, there's some brain fog, but there's other studies relating this connection to cognitive function, neurodegenerative diseases like Parkinson's disease, as we saw this morning, and also mood related to anxiety, depression, and even how a person interacts socially. And so there are many ways, and this is where the microbiome gets into this connection between the gut and the brain. As we know, the microbiome releases many different products, metabolites, proteins that can influence this dynamic communication, and primarily through three pathways that have been identified: neuronal pathway, immunological, and endocrine. So this could be top-down and bottom-up, but for example, the vagal nerve that runs as a highway speed communication between the brain and the gut have sensors to several metabolites produced by your gut bacteria, and can also sense status of populations there. In turn, some microbiota can release certain precursors for neurotransmitters and other molecules that through the circulation can influence the brain, that can cross the blood-brain barrier. There are other things, for example, some microbiota may influence the endocrine system. And when this is dysregulated, it could trigger some disturbances in the lining of the gut. And these could, could be related to irritable bowel syndrome. So there's a lot of mechanisms through which the microbiome and related metabolites could impact this dynamic communication. So going more towards what do we know about this interaction and how it affects neurological disorders, as we saw this morning, elegantly presented this notion that is quite important for Parkinson's disease, it seems. And this comes mainly from animal models. There's this theory where perhaps the disease could start in the gut or the brain first. And in the case of the gut, there's some experimental models showing that bacteria produce some metabolites that can trigger the misfolding of certain proteins like alpha-synuclein. And these can seed and pass on this misfolding and be implicated in Parkinson's disease, for example. There are other examples, for example, with autism spectrum disorders, where in mice where they're exposed to infections in utero, the babies later display some autistic behavior in general. And there are other examples, for example, with stroke. There's a lot of literature suggesting that the microbiome may be very important to post-stroke responses. And there's maybe some people working on this right now. And so what about Alzheimer's disease, dementia? There's more work to do in this regard, but there's some studies suggesting that indeed there could be an implication of microbiome in the brain-gut axis. So, and this is preliminary findings. Some things that are out there in humans are small studies, for example, giving some probiotics and seeing some improved cognitive function, but those are very small studies. There are certain pathways that could be affected, for example, metabolite-related amyloid aggregation. We know that certain bacteria can produce beta-amyloid, and also some metabolites can influence the clearance of this protein in the brain. As we saw, microbiome is very influential in the immunological response. So this type of pathway could trigger neuroinflammation and exacerbate any potential disease already in the brain. There's been other reports of dysfunction of the blood-brain barrier, and as I said before, microorganisms can produce neurotransmitters, and so if that is added up to the brain, what the brain produces, this could create disbalances. But we don't know much about this. So this study, led by our colleague, Dr. Bernard Fongang at the Biggs Institute, is quite interesting and a first report in this kind of area. And he looked at bacterial signatures related to small vessel disease in the Framingham Heart cohort, which included about 1,000 participants who were middle-aged and cognitively normal. And so what he observed, among many other things, is that the relative abundance of this particular bacteria, Barnicella intestinihomis, was related to markers of small vessel disease. And these markers that we pick are, very sensitive to small vessel disease, early changes in the brain. And not only that, but it was also observed that decreased abundance of this particular bacterium was related to decreased executive function, which is kind of a cool finding because it's hard to find those associations in a sample that is relatively healthy. So more to explore there. And so in that context, these are the aims of our grant supported by TARC. First, we want to identify gut microbiome signatures related to Alzheimer's disease and related endophenotypes. We want to further characterize microbial metabolites in stool and relate that to Alzheimer's disease, related endophenotypes, and assess whether these are mediators in any potential association we see. And lastly, leveraging data collected in TARC, we want to explore what are the risk factors related to these signatures in the cohort. So this is our approach. So first, we contact TARC sites, offering collaboration. We assess whether participants can be contacted, whether a participant will be interested. And once all the regulatory affairs are in place, we can move to the contact of participants. We mail them information about the study. And then if they're interested, we perform a quick screen. We need to assess whether they've had any recent GI events for sample collection. And of course, we consent the participant. Then we send them a kit for easy stool sample collection with instructions and a return label so that they can send that back to our lab. And once the sample gets to the lab, we process, aliquot, and biobank at the Biggs Institute. In parallel, we also collect other variables and information related to different health variables by phone or over RedCap. So this study is completely remote. They don't need to come to any visit. We hopefully will match other clinical information through other studies. And we've selected the Genotech OmniGene Gut for metagenomics and the OmniMet Gut for metabolomics. And these offer advantages in stabilization of genetic material. They're also flexible with temperature changes, especially in Texas, although we provide ice and all that is needed to keep the sample stable. And it also allows for standard volumetric samples. And we hope that the accessories we include make it user-friendly. So once all this, right now it's being stored at the Biggs Biobank. When we complete enrollment, the idea is that we're going to send this to our colleagues at Baylor for metagenomic sequencing and the other samples to Metabolon for metabolomic profiling. So what is the other information we collect? We assess current living situation, any GI symptoms, vascular risk factors like smoking, BMI, things like that. Relevant medications, that's quite important for analysis. Any new events, neurological diagnosis, etc. And we do perform an ECOG, both for the participant and an informant. And all this is done over the phone, and the ECOG can be done in a REDCap form. And then, of course, as you saw, diet is quite important. So we are doing the diet history questionnaire. This is a standardized food frequency questionnaire that includes over 130 food and beverages, 26 dietary supplements, and it gives information on the frequency and the portion size. And this was developed by the National Cancer Institute based on the Enhanced survey. It's very nice because it integrates information and gives you not only the food intake, but macronutrients, micronutrients. So it's nice, and all this is available in English and Spanish. So it's very, very good for our population. And it also generates a nutrient report that is kind of a return of results to the participant if the participant wants to receive it. And so this is also, again, participants can go to the DHQ3 website, answer the food frequency questionnaire in their own time, and if they prefer, we can just go over that over the phone with our research staff. So right now, we've been collaborating with UT Health San Antonio, Baylor, Texas Dell, and we are right now hopefully starting to wrap up things to begin collection in Texas Tech. We expanded the study at some time to include participants from cohorts that have a similar assessment in cognition and, for example, that use the UDS tree and that have other very interesting neurological outcomes. And these include our South Texas Alzheimer's Disease Research Center and a study focused on vascular contributions to cognitive impairment and dementia called MARC-VCID. So this is our progress so far. We have about 116 participants who have completed the study and about 55 other who are ongoing responding questionnaires and about to return the samples, etc. And so hopefully next time I can present you results about this exciting study, but this will provide critical information and preliminary data for future grants and other opportunities. This is already the case for our colleague Bernard Fongang. He's going to be using this data in his K Award. And hopefully we can build up on that and expand the study to include more participants. And ultimately, we hope that these results can contribute to preventive or therapeutic strategies to halt cognitive decline. There are many ongoing trials around the world, if you can see this picture here, in all kinds of different things, especially GI-related diseases. But there could be developments in prebiotics, probiotics, fecal transplants, and any other kind of engineer personalized medicine. That's the idea. And yes, so hopefully we'll get more information soon. And I want to thank our team. It's amazing. Our faculty, Dr. Godslav, our research staff, and all the TARC collaborators who've been great with us and are part of the study. And we're open. We're still enrolling. Send me an email if you're interested. We hope to close enrollment at the end of the year, and then we'll send out the samples for reading. Okay, that's it. Thank you very much.
Claudia. One thing was, is the microbiome, your gut microbiome, is it related to ancestry for two reasons? One is like how influential is different diets to influence like what your gut microbiome is, and the second is the genetic ancestry of the person like just like population stratification, is there like different microbiomes in different regions, geographic regions of the world? And
Yes, so because microbiome is so dependent on the exposome, you can imagine that the microbiome of an American will be very different from one in Japan. There's been some experimental studies showing that although the microbiome is malleable and you can change it with food, if you do the same over generations, the newest generations will have a harder time changing that microbiome with different diet. And then when you compare the microbiome of hunter-gathering people, you see that there is a completely different and much more diverse microbiome. And these resembles that of some excavations and some archaeological findings. So yes, although it can be changed, it probably has a generational component. There was a GWAS looking at gut microbiome. It did find some genetic associations, but it was not that strong. So primarily it's mostly influenced by the environment.
Any other question? Thank you.
Thank you.
Please welcome Dr. Giuseppe Tosto. Dr. Tosto is an assistant professor of neurology at the Gertrude H. Sergievsky Center and the Taub Institute for Research on Alzheimer's Disease and the Aging Brain at Columbia University. Dr. Tosto is a genetic epidemiologist with funded projects recruiting Hispanic Latino individuals phenotyped for Alzheimer's disease in North, Central, and South America. His other projects focus on polygenic risk scores, disease heterogeneity, and the use of disease endophenotypes such as neuroimaging, CSF, and neuropsychological predictors.
Hello, everyone. Thank you for the invitation. Let me see if yes. This is my first time here at the TARC Symposium. It's pretty exciting. I've been dealing with TARC data for quite some time indirectly. I'm part of the Alzheimer's Disease Genetic Consortium and the Alzheimer's Disease Sequencing Project, which TARC is part of. So our cohorts at Columbia, just like TARC, we like to share data. We are part of this big consortium, which I truly believe in data sharing. So I'm very happy to be here in a place that have the same belief. And then recently, I've been started collaborating more directly with some of the researchers here at TARC, specifically Dr. Barber and Dr. Phillips. Has been a wonderful collaboration. I don't know what I enjoyed more, working with these wonderful people in Texas or not working with people in New York. Who knows? I'm joking, sort of. So today I've been talking about APOE. I don't know who is familiar with genetics, with APOE. Most of the time, APOE seems kind of boring because everybody knows about APOE, the association. It's pretty established. It's been decades that this gene has been known associated with Alzheimer's disease. But I'll try to raise your attention anyway. I'd like to start with my with my group. Usually, you can see these pictures at the end of the presentation. I like to put it at the very beginning because they are really the people that do most of the work. It's a very diverse group, and diversity will be, kind of like the magic word in my presentation. As you can see, we have our clinical coordinators from Dominican Republic. We have scientists from India, from China, from Peru. I'm European. So when we have our, weekly meeting, at the end of the meeting, sometimes no one has really understood what we have been talking to each other. There are some, serious accents going on. As I said, the diversity is kind of like the magic word, and it's particularly important in genetic. It's pretty well established that the existing pool of genetic data is dominated by European descent population, right? So this is pretty popular website that monitor, how diverse, what's the composition of the available data. And as you can see from the pink, most are coming from cohorts that recruit European descent individuals. On the right panel, you see that over the years, there is an uptick in studies recruiting non-European, so non-white individuals, but the scenario is far from being well balanced. Many things are changing, though. Here is another view of the existing data by condition. So you can see metabolic disorders, cardiovascular diseases, and then everything, again, is dominated by European descent population. Probably neurological disorders it's the condition where there is more diversity. You can see by the color. But again, as I said multiple times, there is a lot to do still. Things are changing, fortunately. I like to always give this kind of example. This is the famous human genome. And this is the original project, and as you can see, the composition again was dominated by genetic material from European samples. Now we have a new pan-genome reference project that embrace truly diversity. So right now there are 47 individuals of diverse ancestry. And pretty soon, the number will go up to 350. So you clearly see from these figures the great improvement done by this reference panel. Oh, by the way, through my presentation, you can see a QR code. So in case you want to have additional details, just use the QR code. I'm now obsessed with this new thing, like a good boomer. So I'll try to stay on each slide, long enough so you can, take a shot. Speaking of diversity, this is another pretty striking example. The genome AD, it's for us geneticists, it's something we deal with on a daily basis. The new release, the fourth release, has now an impressive number of exomes and genomes, over 700,000 exomes, over 70,000 genome, and it's incredibly larger than the release number 2 and release number 3 combined. And this time, the number of non-white individuals has grown massively. Over 120K individuals are of non-European genetic ancestry. So again, increasing diversity, increasing not only, it's not just a matter of sample size, even though in genetics, big data sets are very valuable. But as you can see from this little graphic here, despite the number of Europeans being still massively higher than the other population, the number of, you can see on the here on the right side, the number of non-synonymous variants, so variants that we genetic variants that have potentially an effect on our system. The number, it's really driven by this little portion here. And the number provided by this big chunk here, it's pretty small. So numbers don't always match with the quality of information that are provided. And this is, again, I hope by now I have convinced you of how important it is to have all the colors, all the rainbow. This is a slide that has been probably most of you have seen, even if you're not working in genetics. Every geneticist roll their eyes every time they see it. It's kind of like an overview of all the different genes associated with Alzheimer. So going into a more specific condition I will be talking about. Pretty much this has been updated to 2018, 2019. At some point I stopped updating the slide because there are more and more genes being discovered every year, every month actually. But one gene is always there, which is ApoE. ApoE either having one allele or two allele of the E4, which is the isoform, which give an increased risk for Alzheimer's disease, right? So ApoE is always there. It's always first in the chart, always winning. And even though people seems to be more interested, in all these little other genes, sometimes with very small effect size. And nothing really, except for a few exceptions, can compare to ApoE, which is still nowadays the strongest genetic risk factor for Alzheimer's disease, the most validated and replicated. In addition to the, again, very famous E4 allele, and then E2, I think you heard about this earlier today, there are ApoE keep providing additional information. We heard earlier before by Professor Duff about the Colombian cohort. This is still related to ApoE, the Christchurch mutation, which provides a protection even in autosomal dominant Alzheimer's disease. So there is a lot still to unpack within the ApoE gene. Moving on to from common variants, so such as ApoE, E4, rare variants are extremely important, and this is why I mentioned earlier about the Genome AD and all these large reference panels that give us a real insight into the highest number of pool of genetic variants possible. Rare variants are very valuable to study even common conditions such as Alzheimer's disease. And this is one of the many recent studies showing how rare variants affect common traits and how aggregating rare variants that singularly has might have little effect sizes, but together they capture way better than common variants the phenotype landscape of, for example, Alzheimer's disease. And this is just to summarize how rare penetrance mutation in genes implicated by the GWAS confer many for larger effect sizes than common variants. And I'll talk a little bit about polygenic risk score later today. We also know that dementia is projected to increase over the next few years. But we also know very well that the projected change across the world is not even. There are certain areas, specifically Africa, the Middle East, and South and Central America, where the increase will be dramatically higher compared to the Western world, so North America or Europe. And so from there, specifically Central America and South America, it's where my group and many other groups are conducting recruitment, as well as TARC. So I'll be talking about Caribbean Hispanics and Peruvians and Mexicans, and obviously Mexican Americans. Hispanics, as I said, are projected to be the fastest growing minority among all the minorities in the United States. And they are also particularly suitable for clinical observational trial, because they are mostly concentrated in few areas. One is New York, where my lab is. Another is obviously Florida, Miami. My main collaborators are there. Obviously Texas, not to mention, and California. This is one of the seminal study from Columbia University now, almost 25 years ago, that showed that Alzheimer's disease has a higher prevalence and incidence in Hispanics and also African American compared to whites, to non-Hispanic white. This is taken from the YCAP study, which is a multi-ethnic ongoing longitudinal study in northern Manhattan. And so I think so far I convinced you about the need to diversify the genetic studies, not only genetic studies for Alzheimer's disease, and why we focus particularly on Hispanics. But what does it mean Hispanic? So it's an umbrella term that encompasses a very heterogeneous number of populations, right? So Hispanics are what we call a mixed population. A mixture, it's the result of previously isolated populations that start inbreeding for various reasons, geographical, social, historical. The modern Hispanics are the result of European conquerors arriving in the Americas, where the Native Americans were, and subsequently the African component was brought in by the slave trade. So what we see now, it's a mosaic of these three ancestral components: European, African, and Native American, right? So this is a seminal study from Moreno Estrada from over 10 years ago. If we plot on a pretty simple component analysis, so a way to visualize the heterogeneity of the genome of different Hispanic populations, in this case we have Mexicans, Colombian, Dominicans, Puerto Ricans, together with the reference populations, so African, European, and Native, you can clearly see it's pretty striking how heterogeneous is their composition, how they're stretched in between these three reference corners, and even very distant from each other. Look, for example, where the Mexicans are located on this principal component graph compared to Puerto Rican or Dominicans, right? So this will introduce my next topic on how bias is to just group such a different pool of populations under one single umbrella term. And this is another way to express what I just told you. This is a simple plot of the proportion of ancestry across Caribbean Hispanics, Mexicans, and three Peruvians population that we'll describe later. You can see the Native American component, which is almost inexistent in Caribbean Hispanics, become predominant, if not the only ancestral component in indigenous population from Peru, and vice versa for the European ancestry. So very well represented in Caribbean Hispanics and almost nothing in indigenous population. Obviously, this is the result of social events. The Caribbean Hispanic population, so the native population from the Caribbean island, were very exposed, they were the first place where the European conquerors arrived, and so they were pretty much wiped out very, very fast. Whereas inland populations, especially population residing on the Andes, so on remote areas, were less protected, and so this is the result, the modern result of this different, very heterogeneous dislocation of the European conquerors and the native populations. So this is another important point. Ancestry is not only relevant globally, so how much African, or how much native a certain individual is, but also locally. So we do know that Alzheimer's disease or any other disease there are specific genes that are particularly relevant. And it matters at that specific location if the ancestral background is coming, for example, from a European ancestor or an African ancestor. This is a study from the Miami group that showed that, again, ApoE, because this is my main topic today, regardless of the isoform, so if an individual is an E4 or an E3 or an E2, if that isoform resides into European block or an African block, so what we call a local ancestry modifies the association between the ApoE and the disease. And speaking of local ancestry, this is a paper that we published a couple of years ago when we studied the association between local ancestry and Alzheimer. This technique is called admixture mapping. So we map the local ancestry and try to find association with our outcome. And we were able to identify, obviously, beside ApoE, another signal on chromosome 22. And then this is exactly what you see in an admixed individual, for example, an Hispanic individuals. This is chromosome 22, and you see this specific region has an increase in cases versus control of a specific ancestry, local ancestry. And under this region, admixture mapping identifies very large section of the genome. We were able to map through sequencing data a specific gene that then we follow up with a lot of functional experiment, and we had a nice paper. Again, exploring Alzheimer's disease in the context of a mixed individual, such as Hispanics, is particularly valuable because we know that the risk of Alzheimer varies across ancestry. For example, this is a pretty recent paper. They studied the prevalence of dementia in a Bolivian indigenous population, and they found extremely low prevalences of dementia, which is exciting. It's super interesting. You would expect that population with low access to healthcare, very different nutritional habits, exposed to a number of diseases, would have worst outcome. Well, that's not the case. especially compared to other indigenous populations, for example, Aboriginal populations. And this is something that we are trying to explore. We still don't have an answer. Since like Native American ancestry, it's somehow protective towards Alzheimer's disease. So this is not the first report. There are other reports, for example, in Colombia, in Brazil, that shows that there is some sort of protection towards Alzheimer's disease. But going back to APOE, now we did a quick tour of the planet. APOE as well differs across populations in terms of frequency, but also effect sizes. You can see here, it's well established that the effect size of APOE for Alzheimer's disease for example, in African Americans or in Africans, it's very low compared to non-Hispanic whites. Whereas in other population, for example, East Asians, here we have Japanese population, the effect is extremely strong. And then we have Hispanics. We have Hispanics, again, using this very wide, vague term that usually provides conflictive results. You can see here the effect size, it's kind of like in between African and European and East Asian, as you would expect, but when you raise to homozygous E4, you kind of have the same odd ratio, which doesn't really make sense, right? We do know about APOE being definitely less impactful for Alzheimer in Hispanics. This is another study that we published a couple of years ago for polygenic rescore. So we tested the classification performances of the polygenic rescore in Hispanics versus what we know in Europeans. And we found pretty good performances, but the reason why I'm using these slides is to show you the contribution of the classification from APOE. So it's a pretty strong area under the curve just for APOE, whereas for the Hispanics, the contribution is pretty lukewarm, right? And so another way to describe what I just said is to just plot side by side the area under the curve, and you see how in Caribbean Hispanics, the contribution of APOE is definitely lower than what you have in non-Hispanic white. Another study from INCA, another conflictive results. In this case, has to be appreciated, the term Hispanic was not used, but it was breaking down by Cuban and Dominicans, all the different population. But if you see the p-values and the odd ratio, all are pretty lukewarm, maybe except for Cubans. In this case, the outcome was not AD-MCI, but still you would expect more robust results. And finally, this is the paper that I want to highlight the most. This has been published a few months ago, so it's extremely fresh out of the oven paper. A lot of the data, some TARC data could be in here. Definitely a lot of data from Colombia and part of the ADSB. As you can see, Hispanics were compared to African Americans, to whites, and to East Asian. And what is clear from this graph and from this table, the association between the E4, E3, or even the protection from E2 is the weakest in Hispanics compared to the other population. It's even weaker than African Americans, which is very surprising, right? It's not what we would expect. And so one of the things that we speculate that this approach is pretty biased. It's biased for the reason that I explained before, so grouping population that are extremely different, extremely heterogeneous, even within the same country, under just one census group, can lead to very conflicting, or I would say biased results, right? And so what we did in collaboration with TARC was to group the highest number of Hispanics that we could, Caribbean Hispanics coming from Colombia. Again, you can see the QR codes, they're back. We have here a YCAP, which is a multi-ethnic study from northern Manhattan, great representation of immigrants from Dominican Republic and Puerto Rico. We have 1066 Dementia Puerto Rico, and we have the FIGA study, which is a study of large multiplex families with multiply affected with AD. And then we have HEMAS, so the Mexican Health and Aging Study, run here in Texas by Rebecca Wong at Texican Medical Branch, Galveston. This study has been going on for over 20 years. You know the deal. It's a great study. Over 20 years follow-up, recruitment in all 32 states of Mexico, plenty of phenotype, biomarkers, and then finally a few years ago we added the genetics. So now we have GWAS on almost everyone, and we're generating whole genome sequencing. You can request the data freely. And then this is what I'm probably most proud of, is the Peruvian study that I've been mentioning here and there in my previous slides, is the GAP study. This has been, it's a recently funded study. We have been recruiting in Lima, in the Andes, Puno and Arequipa, and in La Paz in Bolivia. And this is the wonderful team behind the recruitment. And these are some beautiful photos that actually the interviewers took on site. I was listening to the wonderful presentation from Professor Duff, who used to be at Columbia with me. She probably doesn't remember me, I'm sure. So I was amazed by, yes? Oh, great. Oh, you made my day. I was amazed by virtual reality and, helicopters and, the armed people. We did not have that. We were poor. Actually, the interviewer that took this photo, she was going around Puno on a bike to interview our participants. I also have to say that Peru is a very quiet, very safe country. The worst that can happen is a llama, spitting on your face. But now, so we were poor, now we're less poor because we were funded, again, by NIH. Thank you, NIH. And now we're increasing our recruitment in over 10 sites, all across Peru and Bolivia. And again, you can have more details. So we put together Hispanics, I had to use now air quotation, from Caribbean Hispanics, from Puerto Rico, Dominican Republic, and New York, Mexican from Mexico through HEMAS, Peruvians and Bolivians from GAP, now called GLASS, and finally, I'm not going to describe this, HUBS HD and TARC, Mexican Americans. And this is probably, I could have skipped all the slides before and just show you this. This is an overview of these 4 different population that are making this super population, Hispanic population, either through principal component analysis or ancestry. And just looking at the distribution of the color, right? So the green is an African ancestry. The blue is European ancestry, and red is native ancestry. Just looking at the composition and the proportion, you see how different these 4 superpopulations are, right? There are a lot of colors in my presentation, I know. And here we have the usual 3 reference population: European, African, and Native American. And these are the results. First of all, once again, I want to drive your attention on the composition, the ancestral composition, the ancestry in the populations. So we have European going from almost 60 down to 23 in Peruvians, Native American going on the opposite. But what's, I think, most interesting is the effect of APOE. So I've been talking about how heterogeneous are the results so far. And, well, first of all, we confirmed the strong association between APOE and Alzheimer. That's a given. That was not under discussion. Hopefully, our results said the same. But going from a 2.5 odd ratio, we go up to over 6 in Peruvians. So this tells you again that establishing an association behind this story, there's another story. So there is an extremely heterogeneous effect size between this gene that has been known for decades, and our main outcome. And it kind of makes sense. Again, remember my previous slide, the Peruvians are highly native, and keep in mind that native, evolutionary speaking, are coming from East Asia, right? So over 1,000 and 1,000 years ago, they passed through the Bering and came down to the Americas. And so this high effect size in Peruvians, so in highly native population, kind of resemble what we see in East Asia nowadays. So this strong effect size of APOE, way higher than Europeans. And this is a simple meta-analysis, so not much to see, establishing the effect of APOE across different populations, but again, look at the heterogeneous effect. And we were able to, contrary to previous paper, to confirm the protective effect of E2. So E4 risky, E2 protective. Again, we leverage the longitudinal data that some of these cohorts have. Longitudinal data are wonderful to validate, replicate, corroborate your findings. These are the Caribbean Hispanics from Colombia. And this is a simple Cox regression score table that shows, obviously, that the survival time for E4 carriers is dramatically lower than whoever doesn't carry an E4. And then further stratifying by the 3, so no E4, 1 E4, or homozygous for E4, split even more the 3 curves. I think it's interesting to look at the mid survival, so the 50% survival, the anticipation of almost 10 years for going from a non-carriers to homozygous E4. And then my last slide is, again, going back to the Peruvians. So one of the major arguments for all these study cohort that rely on clinical diagnosis is how good is the diagnosis, right? Especially neuropsychologists here, geriatrician, neurologists, there's always this big question mark. Is this, what you're observing, a true result or just a bias because, your diagnosis is not that good? Keep in mind that a lot of tests that we use for recruitment or even in clinical trials are designed on European populations. So moving to minorities, even more moving outside the United States on another language, pose a lot of methodological issues. And so this is why we, to be OCD and super sure about what we were doing, we employed one of the best biomarker now on the market, so the p-tau 217, and simply check if our cases and our controls, how they were behaving, using these biomarkers. Well, seems like they're behaving pretty well, right? So I don't need to put a statistics or further explanation on this graph. And also using ApoE and checking the level of p-tau in blood also shows you how the separation between each E4 allele becomes very striking. Don't look at the homozygous. Obviously, the number, the Peruvian cohort, it's very small, and so the number of homozygous E4 is extremely small. Therefore, your confidence interval are super wide. But just focusing on the first two groups, you see how you don't even have an overlapping confidence interval. And so finally, this is the acknowledgement for all the people who have been working on this project, these wonderful collaborations, particularly Dr. Barber and Dr. Phillips and Sid O'Brien from Texas. And last, I know, I still have 2 minutes. So I know Texas is wonderful. I've been enjoying the sun, a lot. But if anyone, is out of sunscreen and likes long, gloomy, depressing winters, we're hiring. Thank you.
And there's going to be a ton of questions after such a great talk, so hopefully you can hang around afterwards and address them.
I'm
I
here.
have time for one, and I think you were the first.
Can you please speak in the mic for me, please?
Go up the mic, though. Great job.
So this may be a naive question, but in terms of ApoE, you said the local ancestry of ApoE is important, and in terms of just general ancestry, you said Native American ancestry is protective for dementia. Which do you think is more important? Do you think the local ancestry is really important? So, for instance, if the local ancestry is in ApoE is like a good a protective ancestry, and the general ancestry is not, do you have a comment on
Oh yeah, no, this is a great question. It's not naive at all. No, definitely, I would say local ancestry is the real deal, right? So, because you can be, let's say, 90% African and only 10% European, but if that 10% is localized in, in specific genes that are relevant for your outcome, then your global profile doesn't really matter, right? So so that's my straight answer.
The rest of the genome, the risk.
We need to follow him afterwards.
I'll be here. Not going back to New York.
Please welcome Dr. Joanna Jankowski. Dr. Jankowski holds the Vivian L. Smith Endowed Chair in Neuroscience at Baylor College of Medicine. Her research focuses on understanding factors such as genes and aging that influence Alzheimer's risk, and testing experimental approaches for treatment, including gene therapy. Her work uses genetically engineered mouse models for Alzheimer's disease as a testbed for these studies, and she is recognized for helping to create and characterize several transgenic models of Alzheimer's disease. At her home institution, she teaches extensively and is the Associate Director of Graduate Studies in Neuroscience.
All right, Voice of God is done, so now I can start speaking. My thanks to the organizers for inviting me to speak here today. My thanks to TARC for funding the continuation of the work that you'll hear about. My thanks to all of you for staying through the bitter end. In reward for that, I will try to keep this on time and light. And I will start by saying, let's see, green button did not just work. There we go, green button is working. I Despite the introduction, I'm a pretty simple-minded scientist, and I try to think about our science in the simplest terms possible. I'm going to argue that everything I tell you about today can be basically thought of in terms of what you learned in kindergarten, Lego bricks. I think of A beta monomer as an individual Lego brick. And you can imagine what happens with individual Lego bricks. They stack on top of one another and form what one could think about as a growing amyloid fibril. And if you are lucky, your building blocks come together in something like the fancy moon lander that is on the top of the package of the Lego kit that you just bought for your child. But if you're unlucky, you end up with something like this, an amyloid plaque. If we agree that some part of this process is fundamental to the pathogenesis of Alzheimer's disease, then anything we can do to arrest this process is going to be therapeutically beneficial, or so we would hope. Well, what if you could interfere with that process? What if you could stop those Lego bricks from stacking on top of one another? Could you do that through something that might be a structural variant of a Lego brick or a structural variant of an amyloid beta peptide that shared just enough similarity that it was able to sit down on a growing fibril, but just enough dissimilarity that it would prevent anything else from binding down and thereby stop the process of fibrillization and plaque formation. This isn't a new idea. I am just lifting off what people have been working on, including people like Claudio Soto, for decades. There is a long history of inhibitor peptides that are designed around the fundamental amino acid sequence of amyloid beta, with slight variations in that sequence that introduce three-dimensional structural changes that prevent them from aggregating in a test tube. This was all wonderful until you started thinking about clinically applying those inhibitors. And there, the biochemists who were great at identifying these inhibitor peptides in test tubes ran into two roadblocks. Number one, if you stick naked peptide into the bloodstream, it gets very quickly degraded by proteolytic enzymes. And number two, if you were able to modify that peptide so that it would remain intact in the bloodstream and survive the gauntlet of enzymes, you still have the blood-brain barrier to get across. My addition, my advance on this decades of elegant and really just deep work on the biochemistry and biophysics of amyloid beta aggregation is to understand that we might be able to apply a Trojan horse to get across those two obstacles. Well, there are 4 basic criteria that I think any inhibitor peptide is going to have to meet to make it worth packaging into some sort of a great Trojan horse. One of those is that if we make this structural variant that has slight amino acid variations on standard A beta, it can't self-aggregate. It can't make things worse. Number 2, it's got to do its job. If it is meant to be an inhibitor of the aggregation of wild-type A beta, it's got to be able to do that effectively. Number 3, if we were really lucky, we would find a peptide that might actually be capable, through mechanisms that I don't fully understand yet, of disassembling pre-existing fibrils, and therefore might be beneficial after amyloid onset. And number 4, whatever we produce has to reduce the cytotoxicity of remaining A beta species. So we can't generate an intermediate species that's worse than what we would have had otherwise. I have colleagues who basically say, take nothing on authority. And so we went back to the literature and we tested our own A beta inhibitor candidates before thinking about packaging these into devices that might get across the blood-brain barrier. So we are really lucky to live in an era where really advanced cryo-electron microscopy has given us basically a molecular structure of the amyloid beta fibrils that tells us exactly where every amino acid in amyloid beta is located within a cross-sectional picture of an amyloid beta twisted fibril. It's been known for decades that a core set of residues are critical for this aggregation of amyloid beta into fibrils. And we targeted a set of 3 of those, along with one extra that was slightly outside of that core. And we generated synthetic inhibitor peptides that were produced by a company for us to test in vitro, along with wild-type A-beta-42, and ran through a series of different tests to examine each of those 4 criteria. Number 1, could we show that any inhibitor peptide that we identified as being a good candidate from the literature lacked the ability to self-assemble? So this is a basic assay that's used in aggregation studies. It's called a thioflavin T assay. And what you can see here on the x-axis is time. On the y-axis is the fluorescence of the dye thioflavin T. And what you're studying after adding 3 components, buffer, amyloid beta, and this dye thioflavin T, is the intercalation of thioflavin T into the growing fibrils. As amyloid beta aggregates, you can see that the fluorescence increases. This is what happens with wild-type A-beta-42 in this assay. Now, what happens with any of our inhibitor candidates when we do the same thing with them in vitro? All of them flatline. All 4 or 5 of the candidates that we tested showed no aggregation properties in vitro. Great. Check off number 1. They don't self-aggregate in vitro. But number 2 is the thing that we're really interested in. Sorry, I'm having a little bit of trouble here. Nope, that's why I'm hitting the wrong darn button. That's exactly what they said I would do, too. So second thing, it has to do its job. It has to prevent the aggregation of wild-type A-beta if they are co-aggregated together or co-incubated together in what's called a competition assay. Again, the same thioflavin T assay where we're looking for fluorescence on Y against time on X. This is what happens with wild-type, incubated with wild-type A-beta. This is what happens with our inhibitor candidates incubated with wild-type A-beta. I think you can immediately see that one of them we went, oh my, and that's this guy right here. It made things worse. Knocked out, completely did not meet our criteria. But we have 2 others here, shown in red and blue, that completely, again, not only didn't aggregate themselves, prevented the aggregation of wild-type A-beta in this assay. If we were really lucky, we'd find one that was actually able to disassemble preformed fibrils. So A-beta can be aggregated in a test tube, and then you can incubate it with thioflavin T and buffer. And if you study that over time, you can see here that the level of fluorescence stays stable. The fibrils are basically reasonably stable structures. But through, as I said, mechanisms I don't fully understand, and I would love to talk with people offline about this, if we now incubate with an excess of our inhibitor peptide, a preformed fibril, we now get a decrease in the amount of fluorescence, suggesting that they are disaggregating existing fibrils. Finally, and this is the most challenging to explain of the 4 criteria, can we make sure that we don't generate some sort of an A-beta structure that makes cytotoxicity worse than we would have with just A-beta alone? So this is where we take A-beta that is either monomeric or aggregated in vitro into oligomers, and we then apply them to a cell culture, an N2A cell culture. And we ask, compared to baseline, no peptide exposure, what happens in 24 or 48 hours to cell survivability? Well, if you expose it to wild-type oligomers, you kill cells. If we now aggregate just our inhibitor peptides by themselves, we know they don't aggregate and they don't kill cells by themselves. But here's the key point of this, is what happens when we now co-incubate wild-type A beta with either of our inhibitor peptides during oligomerization. I think this is easiest to look at if you just compare wild-type A beta oligomers by themselves versus wild-type A beta oligomers that were formed in the presence of either of our inhibitors. And as you can see, they don't completely rescue cell survival, but they do improve it significantly. Well, we now have 2 inhibitor peptides that are each slight sequence variants on wild-type A beta that have met all 4 of our criteria. Well, the job now is to get past that roadblock. Well, the ancients did it with a 25-foot-high Trojan horse. I realized that we could do it with a 25 nanometer wide adeno-associated virus. Our challenge then, and I won't go into the details for the sake of time, was to figure out a way to go from the amino acid sequence of our inhibitor into a vector sequence, a DNA sequence, that could be packaged into AAV and effectively deliver our variant peptide into neurons and allow that variant inhibitor, or variant peptide, to be generated by those neurons as processing factories and then secreted extracellularly in the brain so that that inhibitor peptide would then be exposed to the spaces extracellularly where wild-type A beta naturally is forming fibrils and plaques. Well, as many of you in this audience know, you can bypass the blood-brain barrier simply by injecting things like adeno-associated virus with intracranial injection into the brain of adult animals. But it doesn't spread very far. One of the things that's really great about AAV is that if you do this into neonatal animals and inject into the ventricular system of neonatal animals, you can basically fill the entire ventricular system with fluid that contains your virus, and because the ventricular lining hasn't matured yet, and the AAV is so small, it passes through the ventricular lining, transduces neurons widely throughout the brain. And so we began our studies with this approach, which I consider a sledgehammer approach. Again, okay, thank you very much. I was about to say I'm losing my monitor here. So began with what I would consider sledgehammer studies. I am telling you about a physical way of crossing the blood-brain barrier, and we are talking about introducing a virus that secretes an inhibitor peptide from birth. But if we want to do the proof of principle, that's going to be the best way to approach proof of principle. And so what we did was to inject our inhibitor viruses, or inhibitor peptides packaged into virus, into neonatal mice. It then transduced broadly throughout the brain, and the neonatal mice that we used were animals that overexpressed amyloid precursor protein, and which, if we aged until 7.5 months, would develop amyloid beta plaques throughout the cortex. So what happens if we now study, this is an uninjected control here, we can now significantly diminish the formation of amyloid plaques in APP transgenic mice by treating them lifelong with viral vectors. Is this counting up or counting down? Okay, good. I have time. I was like, oh my God, am I at 25 minutes? Are you guys all, okay, fantastic. So we've significantly inhibited the formation of amyloid plaques. Fantastic. We've also successfully inhibited microgliosis in these animals. So one of the things you would worry about in putting a virus into the brain is the possibility of immune reactions to that virus. AAV is supposed to be a low immunogenicity virus, and it is widely used for gene therapy. But take nothing on authority. Check it for yourself. What we did find is that if we stain for microglia using an antibody against IBA1, you can very clearly see the plaque outlines in uninjected controls, but we see very little microglia activation in our virus-injected transgenic animals. And this is shown in brown up here, shown in, poorly in red and green down here. But we also have to check for the immune response in non-transgenic animals to make sure, again, that our virus is not causing an immune reaction that is unwanted. Here we injected into non-transgenic animals, injected at the same time, harvested at the same time. You can see that our AAV delivery seems to be relatively inert, at least for microgliosis. Well, I wouldn't be telling you this story if that were the end of the story with regard to immune reaction. When we next looked at astrocytosis, the story wasn't quite so rosy. So here we are immunostaining for astrocytes in the cortex, and you can see that the amount of astrocytosis around plaques goes down with treatment in APP transgenic mice, where we have reduced plaque load. What wasn't such a happy ending for us, and what was quite surprising, was to find that when we injected the same virus into non-transgenic animals, we actually induced astrocytosis in those animals. What we realized upon co-staining and looking more carefully is that this may be happening in astrocytes that actually pick up and start to express our virus. But was there something that we might be able to do about that? And that's where our TARC funding comes in what I would call viral vectors 2.0. So could we do something about our viral vector design that would allow us to avoid astrocyte transduction? And while we were at it, could we make a few other tweaks that might help us improve the efficacy of delivery of our variant peptides in the brain? This is a linearized version of the sequence of our virus. I won't go into the details because I realize what a broad audience this is, but suffice to say that we made a couple of important design changes. Number one, we changed this piece of the DNA that was packaged into our virus. It's called a promoter. It tells the virus what cells to produce our inhibitor peptide in. The promoter that we started with was quite generic. It's called a CAG promoter, expresses in cells throughout the brain, including astrocytes. So we changed that out to a neuron-specific promoter called the human synapsin promoter. While we were at it, we changed a few other pieces of our viral vector design, including what's called a signal peptide that would increase delivery of our mini gene to the cell membrane, and we changed the transmembrane domain that followed our variant sequence so that we could increase gamma secretase cleavage and hopefully improve release of our variant peptide from any of the neurons that were producing it. Well, it worked. So our new A-beta construct actually did dramatically improve the delivery of variant A-beta and diminished plaque load in transgenic mice. So number one, what do we do? Old versus new AAV constructs, we inject again into young mice, and then we harvest out very quickly. These are young mice that don't overexpress APP, so all we detect when we do some sort of a biochemical assay, an ELISA for A-beta, is just our virus-derived peptide. You can see here, compared to the old virus at low and high titers, the new version whompingly beats that. It's like tenfold greater production of our variant peptide being produced in the brains of our mice. And we know it is now neuron-specific, although I won't show that today. What was especially nice for us, and this is data that my student got something like 2 weeks ago, and she is just elated over, was that when we now test this new variant construct in a more aggressive transgenic model of APP overexpression called the 5X FAD, when we inject into young animals, harvest at an age when we know they would have plaque formation, you can see the plaques in an untreated animal are present throughout the cortex and the subiculum. But when we inject these animals with our new AAV construct, we can largely suppress the formation of plaques, even in this mouse model that is more aggressive than our first proof of principle model was. All right, if you maintain a dose of healthy cynicism, you should be looking at me and going, you're injecting a needle into the brains of babies. Like that is it. That's not a clinically relevant gene therapy for Alzheimer's disease. Luckily, people have thought about this, and there are other ways to use AAV that will effectively cross the blood-brain barrier. So what we are working with now are different viral serotypes that will allow us to inject the virus peripherally into the bloodstream, and those viral serotypes have been engineered to themselves cross the blood-brain barrier without any sort of physical needle going into the brain. The second thing you should be saying is, nobody wants to get viral gene therapy before they've been diagnosed with even the risk of developing this disease. And so the next thing we have to do with our tests is to move into animal models that already have amyloid plaques and find out whether or not we can move the needle on it, if nothing else, the formation of further pathology if we treat after onset. And if we're really lucky, some of those inhibitor peptides I told you about seemed, at least in a test tube, to destabilize pre-existing fibrils. So one of the other questions we want to ask is what happens relative to the age at which they were injected? Do we move the needle backwards in time? And if we're lucky next year, I'll have more data to share with you on that project. The other thing you should be asking is amyloid beta, it's old hat, nothing to do here. We've got antibodies for it, that's done and dusted, nothing to see here. You should be asking, can you do the same thing for tau? And that was what we asked as well. And I will admit that I, for years, stayed away from tau because it intimidates me. I have the utmost respect for Karen and the sort of work she has done, the utmost respect for the sort of work that Mark Diamond has done in these areas. Tau, 6 isoforms. Those isoforms can aggregate into a huge range of different three-dimensional structures depending on what disease you get and what cell type the aggregate forms in. So for years, I had just stayed away from this because I think about things in Lego bricks. People have thought about this strategy for tau previously as well. And so there are plenty of people who have been working in vitro in test tubes to use the same structural strategy against tau aggregation based on slight sequence variants. These are largely targeted against the repeat domains that Karen talked about earlier. These are the microtubule binding domains that are critical for the aggregation of tau. And the inhibitors that have been designed are generally short peptides targeting 2 key repeat domains, repeat domain 2, repeat domain 3. The other challenge of working with tau compared to working with amyloid beta is the range of substrates that can be tested in vitro to determine whether you have efficacy for preventing aggregation or not. Unlike A-beta, where you have a 42 amino acid peptide, or maybe if you're going to get brave, you also throw in the A-beta 40 structure. Tau has a whole bunch of them. So tau monomeric substrates vary from this guy here called PHF6, which is literally 6 amino acids that can be forced to aggregate in vitro. Again, if you throw in buffer, something to promote the aggregation like heparin, thioflavin T in that monomer. But there are also 4 repeat tau structures and 3 repeat tau structures, and then you can also get really fancy and study the whole protein. with mutations. And all of these have been studied in vitro and make tau far more challenging than amyloid beta for these sorts of studies. I was really lucky, and I will thank him at the end, to have phenomenally talented biochemists come to the lab a few years ago. And what he was able to do was to mine the literature for those 11 candidates, synthesize those 4 different monomers, and test all 11 inhibitor candidates against all 4 monomeric substrates in that thioflavin T assay for competition against the formation of tau fibrils with inhibitor or tau alone. What I show you here, again, just remember these are the inhibitor candidates we looked at. I will show you the data for the most challenging of these substrates, the full-length tau substrate encoding, full-length tau isoform encoding the P301S mutation. And what he was able to show was that he identified 2 candidates where these candidates don't completely flatline the formation of tau fibrils, again looking at thioflavin T fluorescence over time. This is tau alone, and this is tau plus inhibitor. You can see that it's slowed and it's lowered. This one's accelerated but lowered. Our challenge now is to move these in vivo. I chose to show you this substrate because it's the one that's present in one of the most commonly used tau transgenic animals. So our goal now is to test these inhibitors both in FRET cells and then move into transgenic mice once we tackle the challenges of vectorizing these 2 particular inhibitors. So I'm glad to see that that is the time counting down, because boy, was that worrying me at first. I am wrapping up, I hope, smack bang on time with a few takeaways from our journey. Number 1, everything you need to know to be a full professor working at the forefront of Alzheimer's disease, you actually may have learned in kindergarten. And the second thing is that every time nature throws a roadblock in your way, it's probably also figured out a solution to get around that. And so I think our challenge as scientists will be in future to harness those solutions and use them as best we can to minimize the consequences of disease. I will end by thanking two people who were critical to this work. So number 1, Kyung-Wong Park, who came into the lab with the concept of your enemy's enemy is your friend. And that was how he thought about this story of using an A-beta inhibitor to prevent aggregation of A-beta. He was the one who really brought the idea to us, and I was the one who brought the viral gene therapy idea to him, and together we made a pretty fantastic team to advance this science. Second person I absolutely have to recognize is Nico Mendez, who came to my lab and did all of the tau studies, and it was only through his skills in tau protein biochemistry that we were able to do that work funded by TARC. And I'm delighted to say now he is a first-year graduate student in the Baylor Neuroscience graduate program. They were aided by several talented graduate students and postdocs, and of course, none of this is possible without funding. Thank you very much.
Since I have the microphone, I will ask the first question. So, well, very, very nice talk, Joanna. So, for the study that you did in cells and in the mice, the paradigm was kind of similar. So you started with the combination of peptides and you kind of inhibited the aggregation before testing it. My question is, what happened with the disaggregation? Because if you are disaggregating the fibers, something is happening, you have a residue, you know. My worry is that if that residue will be more toxic oligomers that you are forming. So have you characterized?
We haven't. That's a great idea. We haven't done it. What we had hoped to do through NIH funding was to do that experiment and then do electron microscopy to see what we were forming. We got a little stuck on actually getting synthetic peptide that was intact. So we haven't completed that yet, but we absolutely need to know that.
Yeah. Okay, thank you. Questions?
Hi there. That was a great talk. I had a couple of questions about your animal treatment strategies. So, did you incorporate any cognitive testing into that?
Thank you for that question. You might be planted, and I will pay you later. No, we didn't. The reason we didn't in those studies was because we stuck a needle into the brain when they were neonatal mice, and that's just not fantastic in terms of circuit development, and most of the animals are fine. Injecting virus of any sort, even if it is carrying just inert virus, will lead to cognitive effects compared to uninjected animals. We do have plans to try that and see what we get if we compare to a control virus that just expresses a fluorescent protein, knowing that both sets of animals will probably be impaired compared to a wild-type animal. The place that I am really interested in doing cognitive testing will be in the interventional studies, where we inject the virus into the bloodstream, and we know that we haven't just stuck a needle into the brain. And there I think we're gonna have a better chance of having informative cognitive behaviors.
All right, thank you very much.
Please welcome back the Scientific Director of TARC, Dr. Monroe Cullum.
Well, you've hung in there through the whole day. I personally think the talks have been fantastic. I have learned a lot, which you don't often say if coming away from a scientific presentation day. But the last thing that we have is Dr. Morales is going to announce the poster winners. So if we had a drum, we would start a drum roll. But Rodrigo, take it away.
All right. So we have 4 winners this year. I will name them. They are not in order of first, second, third, so they are all winners. And please, you can wait, you can stand. Please don't come. We don't have nothing to give you now. We will email you, and we will give you your award later. So, all right. The first winner is poster number 12. Deep learning reveals disease-specific signatures of white matter pathology in tauopathies. The presenting author was Charles White from UT Southwestern.
There he is.
There. Second winner was poster 24. Microglial endocytosis of tau aggregates is dependent on actin dynamic and heparan sulfate proteoglycans. So the presenter is Christian Falcon from UT Health San Antonio. Third, poster number 35. Changes of tRNA-derived fragments by Alzheimer's disease in blood serum. Presenter Ernesto Miranda Morales from UTMB Galveston. And the last one is poster number 41, quantification of CSBD lesions using ex vivo MRI in Alzheimer's disease and related dementias. Presenting author, Mariam Modjavai from UT Health San Antonio.
Well, with that concludes our TARC program. I don't know if we have the dates up for next year. There we go. Mark your calendars now. We're going to be back here in full force, and we'll try to at least meet the bar we set this time. But thank you all for coming. It was great seeing everybody, and carry on.